Hey everyone, thanks for the opportunity to present at this forum. I'm Priya Desai. I'm the manager for biomedical informatics, research and development here at TDS. I was hoping to give you guys a quick overview of the STARR DataLake and in particular focus on our two data models that the STARR DataLake supports, STRIDE and OMOP. Here at Research IT, we really take pride in thinking of ourselves as stewards of Stanford's healthcare data. To us, that means management and oversight of Stanford's clinical healthcare data assets to help provide you, the researcher, with high quality data that's both easily accessible and accessible in a consistent manner. As you know, a lot of data from different sources like EHR, radiology, pathology is being generated at the Stanford hospitals and clinics. And all of this can be used to improve patient care and advance scientific discovery. On the left-hand side are listed some of these data sources. So for example, HL7, Epic Clarity, which is basically the EHR records, PACS radiology, bedside waveform, etc. Currently, a lot of this data exists in silos. Our goal is to break down these silos and aggregate this data for research. That is really the vision for STARR. STARR stands for Stanford Medicine Research Data Repository. And the goal for STARR is to create a single integrated data lake containing clinical data of different modalities and to bring this clinical data from its raw state to an analysis ready state and eventually to make this data self-service. STARR is growing to support AI use cases and is bringing in more and more different types of hospital data. It has both structured and unstructured, raw and analysis-ready data. Today, I wanted to walk you through a couple of different data models that the STARR supports for EHR data. The two data models being STRIDE and STARR OMOP. The STRIDE data model, which may be familiar to many of you, has actually supported the Stanford community since its launch in 2008. It is the Clarity EHR data in the STRIDE data format. It's very popular among our researchers and in 2020 alone has helped almost 700 researchers identify cohorts and over 500 researchers have used the chart review tool. This data model is basically homegrown and the data model is closest to our raw hospital Clarity data. STARR OMOP is the new kid on the block. It's basically the same Clarity EHR data, but with the OMOP common data model. OMOP stands for Observational Medical Outcomes Partnership, and this data model is maintained by the Odyssey Consortium. Odyssey is an international interdisciplinary collaboration of researchers whose goal is to create open source solutions for large-scale healthcare analytics. All of these solutions are open source. At this point, we have about 125 users for STARR OMOP and almost 60 researchers who have attended our tutorials. So you may ask, why do we need a common data model? Well, no single observational database can capture all clinical events equally well. Research results from many disparate data sources often need to be compared and contrasted, and we often need to analyze multiple data sources concurrently. Data often needs to be harmonized, and all of this is really hard with location-specific data or a location-specific data model. A CDM basically standardizes the format and content of observational data, so common software applications and tools can be applied across data sets from different healthcare organizations. With the use of data mining using electronic medical health records growing so rapidly, with the use of AI and ML, in all aspects of healthcare like diagnosis, disease forecasting, etc., becoming more and more ubiquitous, incorporating a common data model was really important so that Stanford can continue to be a pioneer in biomedical informatics. So what is the OMOP CDM? The OMOP Common Data Model basically allows for a systematic analysis of disparate observational databases. The concept behind this approach is to transform data contained within those databases into a common format, so a common data model, as well as a common representation of vocabularies and terminology. So, well, what is in our OMOP? With our OMOP, you would actually get access to Atlas. Atlas is a free, publicly available, web-based tool developed by the Odyssey community to explore the structured part of the OMOP CDM. Our installation of Atlas allows you to create cohorts, define phenotypes, run characterization studies, as well as network studies. It's really a cohort tool and so much more. You also would get access to de-identified EHR data, both structured and the clinical notes. This is available to all Stanford researchers pre-IRB via the HIPAA-compliant platform, Nero. And to be really honest, STARR OMOP plus Nero is biomedical informatics on steroids. STARR OMOP is actually hosted on BigQuery, so you can really leverage the power of BigQuery, and this is particularly suited for AI and ML applications. As I mentioned before, this data model is really suitable for phenotyping. We refresh our data weekly, and we have a really solid model for user support, including office hours, a Slack channel, and tutorials. Well, you may say, why should I care? Well, OMOP really allows you to pursue cross-institutional collaborations. It will allow you to really pursue more population health research questions, because you can create cohort definitions, share them with your colleagues in different institutions, and really participate in reproducible research. Stanford first participated in the Odyssey community in the PNAS study in 2016. This was actually an international study with 11 data sources from 4 countries, including EHR records and administrative claims data on 250 million patients. All of this data was mapped to common data standards. Patient privacy was maintained by using a distributed model, and the results were aggregated centrally. Treatment pathways were elucidated for type 2 diabetes mellitus, hypertension, and depression. And this study showed that while the world is moving towards a more consistent therapy over time across diseases and across locations, there is still a significant heterogeneity that remains among sources and really pointing towards challenges in trying to generalize clinical trial results. It was also one of the first studies to demonstrate that large-scale international observational research is feasible. Since then, there have been many more network studies. Very high profile among the recent ones is Charybdis. Charybdis stands for Characterizing Health-Associated Risks and Your Baseline Disease in SARS-CoV-2. There are more COVID-related studies going on across multiple countries, right from studies on the safety of hydrochloroquine to association studies like association of obesity with the severity of COVID symptoms. And none of this would have been possible with a localized data model. It is this common data format that really makes this sort of research possible. Finally, here's a list of STARR resources and links that you may find useful to understand and evaluate whether STARR OMOP data may be useful for your research. And please, if you have any questions, I'm just an email away. So thank you.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.